Bismillahirrahmanirrahim. We start another paper of November 23, paper 4.1, and this is the A2 syllabus, and we're going to do these papers now, and inshallah we're going to finish the 3 paper 4s and the 3 paper 5s for the November 23 so that you can be helped through your exams. As you all know, this paper is for 2 hours, and this covers the 8 chapters of the A2 syllabus, which we are going to be talking about, and the questions which we are going to handle. Now you can see the first question is about on photosynthesis, and usually the first question is a little easier just to break the ice. Chloroplast carry out photosynthesis. Figure 1.1 shows some structural features of a chloroplast and some processes that occur within it. So they've shown you CO2, Calvin cycle product. You know the Calvin cycle takes place in the stroma. Chloroplast, remember ST, stroma. Mitochondria has the matrix, M mitochondria, M matrix. Don't confuse these two. And then they've shown you light coming from here, and then they've shown you product B, H2O, and then product B. Then they've shown you components A make up the granum, and then there's a chloroplast envelope, and there's a product D. So identify the structure labeled A. Now, as we all know, that is the thylakoid and a stack of thylakoid. So you have to get the spellings right. So thylakoid got you a mark, THYLAKOID. You could have said thylakoid or you could have said thylakoids. It's fine. Now it says explain how the structure and appearance of the granum and the components labeled A relate to their function. So structure and appearance and relate to their function. So you've got to talk something about the structure and the appearance. So usually some of you don't read the question clearly. I've been checking the mock exams and I get this very often, in which I see students not reading the question properly and then giving me a sort of a compromised answer, and then I can't give you marks for it. So they would be now, of course, how would we mark this? And this is for 4 marks. It's a lot of marks. So there will be a 1 mark for the description and 1 for the linked function. Like for instance, what could you say? You could have said stacked. The thylakoids are stacked, so this gives a large surface area. You see the thylakoids are stacked like this. And so they give a very large surface area because there are many of them. So why? What is the function of this? The function is to increase light absorption. So light can be absorbed. To increase light absorption. Then the other description you could have said, the thylakoid membrane has photosystems. You know, in the thylakoid, there is the PS2 and the PS1. And then there is the NADP reductase. So PS2, PS1 means what? Then you say thylakoid membrane has pigments, or you can say photosystems. And then what does it do? What is the function of this photosystem? What is going to happen here? Light dependent reaction is going to take place here. So the light dependent reaction is going to take place here. Then we have this thylakoid lumen, the space here. What is the function of the space? Hydrogen ions are pumped in here. So the hydrogen ion concentration increases in this space. So why? Because then it's going to pass through the ATP synthase and ATP is going to be made. That's what the light dependent reaction does. It results in the formation of ATP and NADPH. So then the description appears green under the microscope. Why does it appear green under the microscope? That means that green is reflected. So what is the function? Chlorophyll absorbs red and blue light. So the green is reflected, that's why it looks green. And the chlorophyll absorbs red and blue light. So you realize how it said how the structure and appearance, so the green looks green on the microscope. Structure and appearance of the granum and the components labeled A relate to their function. So you have to be looking at this diagram. This diagram here, this part. How the structure and appearance of the granum is labeled A relate to their function. Now you can see that how I've marked them. You see the points are the marking scheme points are 8. D1 means the first description. Then F2 is the second mark scheme point. So F2 is the reason, the function of it. So D is for description and F is for function. So if you said stacked and then you said this increases light absorption, well, that gets you 2 marks because one is the description and one is the function. Then the second description is thylakoid membrane has photosystems. So that is the third mark scheme point. Then the fourth mark scheme point is light dependent reaction takes place here and makes ATP. Then the fifth mark scheme point is thylakoid has a lumen or a space. And this happens because a lot of high hydrogen ion concentration is going to occur here. Hydrogen ions are going to pump into the space and then they flow through the ATP synthase. I'm just going to show you a diagram of that. And then we said another description is that they appear green. When they appear green, what does that mean? It means green light is reflected. So what does it absorb? It absorbs red and blue light. So these were the different description and function points. Everybody has to be very clear on them. If something is not clear, please read up from the book. And you can always write in the comments and send me a WhatsApp message so that I'll be giving you my phone number at the end of this video. B1, identify the metabolic pathway labeled cycle C. What is that cycle? It's the Calvin cycle. So whether you wrote cycle or not didn't matter, Calvin cycle. Then it says explain why pathway C is described as a cycle. Why is it called a cycle? No start and end point. All molecules are intermediate, present all the time. Ribulose bisphosphate is regenerated. Ribulose bisphosphate is regenerated. And if you gave me the numerical detail of that, then you get a mark for that. Then identify the products of photosynthesis labeled B and D. What was B? B was oxygen. What was D? D was sugar or a hexose or a glucose. You could have said sugar, you could have said hexose, you could have said glucose, you could have said triose, phosphate, carbohydrate, starch, any of those would get you a mark. So there were 2 marks for this. One was for oxygen and the other is for either one of these. Then it says suggest and explain the importance of glucose and the product labeled B in figure 1.1 to ecosystems. Well, that was very simple. Importance of glucose and the product labeled B, there was oxygen. So oxygen would be linked to aerobic respiration and glucose is the source of chemical energy or it is a food. And this is the reason why energy can flow through food chains. If we didn't have plants, there'd be no primary consumers. So there'd be no primary consumers, there'd be no secondary consumers. So the glucose is the chemical energy. So chemical energy which is used for aerobic respiration or you can say in oxidative phosphorylation. And then this allows the energy flow through food chains. You eat an apple, you eat a piece of chicken. What did the chicken have? Chicken had grains. So what you have to understand is how is it going to be? So you have to give me the answer to this concerning the importance of glucose and in importance of it circulating in the ecosystems. Aerobic respiration, source of chemical energy, energy flow through ecosystems. 3 marks. There were only 3 points. You had to give me all these 3 points to get your 3 out of 3. Like you'd have a beginning and an end, but here it is no, there's a cycle. So all molecules are present all the time. So constantly RUBP is being regenerated and carbon fixed, and the same story keeps on occurring all the time. That's what happens in the Krebs cycle, Calvin cycle. These are the cycles that you study. So no start and end point, all molecules present all the time, and RUBP is regenerated. There's not a start and an end, but it is continuously being recycled. The whole of the process is occurring again and again and again and again. Now coming to question number 2. Let's look at question number 2. Biodiversity can be assessed at 3 different levels. I always say paper 4 is something which you need to be very careful in reading your questions. One of these is the genetic variation within each species. What is the variation between human beings? We are different blood groups. I'm blood group B, you are blood group A, somebody is O, somebody is AB. So at 3 levels, one of these is genetic variation. Outline 2 other levels at which biodiversity can be measured. Number of range of different habitats, number of species and their relative abundance. They can be a forest, they can be an ocean, they can be a desert. So number of different habitats or a range of different ecosystems or habitats. Then each habitat, like for instance in a forest, there are 100 different types of trees, and 100 different types of trees and their relative abundance, meaning type A, there are only 2 trees, but type B has 90 trees. Type C has only 5 trees. Type D has 15 trees. So number of species and their relative abundance. Please understand this. Outline 2 other levels at which biodiversity can be measured. This is actually a syllabus point. So number of habitats and number of species and their relative abundance. You see, they told you one thing, genetic variation within each species. Okay, so in this forest, there are only pine trees or mango trees or apple trees. Let me give you things which you can relate to. So what is the difference? We've got some green apple trees and some red apple trees, and we've got these very different types of apples. There are about 100 trees, but they're all apple trees. But there is a genetic variation within the apple trees. Some apple trees are green, some give us a red, some give us a yellow apple. So genetic variation within each species. To calculate the genetic variation that exists within a species, scientists obtain DNA sequences from many individuals of one species. Count the number of nucleotides that differ when the sequences of 2 individuals are compared. Human being A, human being B. Repeat this with different pairs of individuals. This allows scientists to calculate the mean number of differences at every nucleotide position. Along the sequence, mean number of nucleotide differences per site. Explain why scientists use databases and computers to calculate the mean number of nucleotide differences per site. Why do we do that? Why scientists use databases? To store many genomes so that we know the genome or the DNA sequences in a white rabbit, in a black rabbit, in a gray rabbit. And then to compare the sequences. We can compare the sequence of a hemoglobin molecule from a human being or from a chimpanzee. Hemoglobin is a protein. What is the difference in the DNA sequence? So what is the difference in the amino acid and the primary structure of that protein? And then to process or analyze large quantities of data, because computers can process and analyze large quantities of data. Then to share this data which we get from others and then elsewhere, to share the access to the data with other people. And of course the computer, when the computer does it, it saves time. So it's much faster than a human being doing it. Computers are more complex, and they can store a lot of data and they can process a lot of data. So we can say, okay, the hemoglobin, we put all the data in it and the computer then gives us all the answers. So this is of course the reason why we have databases, and all this is a part of the syllabus at this point. Now you can read through the answers. To store many DNA nucleotide sequences, to compare sequences, to process large quantities of data, to share this information with others, and to save time. Is much a faster way of doing all this. Now coming to the C part of the question, table 2.1 shows the mean number of nucleotide differences per site of some species. So Drosophila fruit fly 0.0087, mean number of nucleotide differences per site. Anopheles 0.30301, Plasmodium 0.0015, Z-maize 0.0139. So all this information is given to you. You better have a look at it. Then it says state the genus name of the species that shows the most genetic variation. That's Anopheles. Why Anopheles? Look at the table. State how many kingdoms of organisms are represented in table 2.1. There are 3. Let's look at the table again. So if you look at, what did it say? Anopheles has 0.30301 differences. Then what is this? This is a fruit fly, and Anopheles is a mosquito. So this is Animalia. Plasmodium is the malarial pathogen, is a Protoctista, and this is Plantae. So 3 kingdoms. This is plants. This is a protoctist because it's a pelasmodium, it's a single unicellular. And these two are insects, so this comes under Animalia. So three kingdoms have been represented. So that is why we had this answer here. Then it says part D, genetic variation is considered important in the conservation of species. Low genetic variation is assumed to decrease the chance of the long-term survival of a species. Give reasons why low genetic variation may decrease the long-term survival of a species. Understand this, why? We have about 100 lions, but there's a very low genetic variation because these 100 lions are originally from only 10 lions which were left. So they are less able to adapt. Little variation for selection to act on. What if there is a drought and there's less vegetation? Well, most of them will die. Little variation for selection to act on. So they have lesser useful beneficial alleles. All could be killed by the same disease or by the same selection pressure. And if due to a small population size, this decreases the species survival chance. I just told you that there are only 100 left. So if there is a drought, there's less vegetation. Less vegetation means less deer. Less deer means less food for the lions or the tigers. So while low genetic variation may decrease the long-term survival of a species, because there's not a lot of variation. I can give you another example of trees, oak trees. They have three types of roots: short roots, medium roots, and long roots. Now, if we only have the short and the long roots, then if there is a drought, many years there's no rain, well, the short root trees will die. So you understand that is that genetic variation will result in decrease in the long-term survival. So now the wordings of it, less able to adapt, little variation for selection to act on, fewer useful alleles, all could be killed by the same disease. Why wasn't everybody killed by COVID? Because there's a large variation in the human population which lives on this face of this earth. COVID didn't kill everybody. COVID killed few people. But it didn't kill everybody. Why? Because there's a lot of genetic variation among the people living in Pakistan, in India, in Bangladesh, in America, in Australia, in UK. Small population size, this decreases species survival chance. Then we go on to figure 2.1 shows how the International Union for the Conservation of Nature categorized species according to their conservation status. Common species with the lowest conservation status, least risk of extinction, are categorized as least concern, which is LC. Extinct EX, extinct in the wild EW, critically endangered CR, endangered EN, vulnerable VU, near threatened NT, least concern LC. So the increased risk of extinction is these ones. These are the ones which have the highest to get extinct very soon. Question 2D states that low genetic variation is assumed to decrease the chance of the long-term survival of a species. Predict the relationship between genetic variation and conservation status if this assumption is true. So in 2D we talked about this. 2D we talked about this here. Give reasons why low genetic variation may decrease the long-term survival of a species. So predict the relationship between genetic variation and conservation status. Naturally, if there is low genetic variation, means there is a high chance of extinction. The risk of extinction is very, very high. You see what was happening here. These the ones which are getting extinct are because there are only a few left, maybe 10 left or 20 left of a certain species, and there is very less genetic variation amongst them. So the lower the genetic variation means higher the risk of extinction or higher the conservation status. It goes up here like this, it's going up here. This is higher. This is higher. So lower genetic variation means higher conservation status or higher vulnerability, or you could have said higher risk of extinction. Some of you know it, but you don't know how to write it. Your English is, sorry, pathetic. So lower genetic variation means higher risk of extinction. Then we come to another part of this question. Figure 2.2 shows the mean number of nucleotide differences per site of some species and the subspecies of mammals and their conservation status. So common minke whale seems to have less mean number of, because what do we have on the x-axis is the mean number of nucleotide differences per site into 10 raised to the power minus 2. So chimpanzee seems to have the highest, and then after that we have the gorilla, and then we have the wolf, and then we have the brown rat and the giant panda, and then we have the lion and the common minke whale. But then look at their these things. Look at their gradation key. This is the least concern. So these are the least concern. LC, LC, LC. But they have a lot of genetic. But the wolf has a lot of genetic variation. But it has least concern. Okay, fine. But the common minke whale has very little genetic variation, but it is least concern. Then the lion is vulnerable. So we have these are the vulnerable, VU, these are the vulnerable ones. The giant panda and the lion, and they both vary in the nucleotide sequence. So there's a lot of genetic variation. Now the chimpanzee is endangered, but it has a lot of genetic variation here. This is the chimpanzee. And the gorilla, which has a decently genetic variation, but is critically CR. So you read that from the previous question. Now let's look at what is the question they're asking. But you take your time and look at this table and then figure this out because it's 3 marks. Assess whether the data in figure 2.2 provides support for the prediction you made in 2D2. What is the prediction you made? More the genetic variation or less the genetic variation means higher conservation status, which meant the other way around is more genetic variation, so there's less threat to it, there's least concern. But here it's all gone topsy-turvy, it's all gone weird. Chimpanzee has highest genetic variation, but it is endangered. Minke whale has lowest genetic variation, but is least concern. Genetic variation is same, but conservation status is different. Where was the genetic variation the same? You see here, which was genetic variation was the same? The lion and the minke whale have the genetic variation nearly the same, but one is vulnerable, one is least concern. Then you can see the wolf and the giant panda were nearly the same, genetic variation, nearly the same. This is here and this is here. But then look at their status, they were different. So you gave me an example of this, you got a mark for that. Gorilla is most critically endangered, but has a high genetic variation. It has a high genetic variation. But then, of course, we can also say this is only limited to, the data is limited only to 7 species. We didn't have a data for a large number of species. It's only 7. 1, 2, 3, 4, 5, 6, 7. So the data is limited to only 7 species. So that is the sort of thing which you can write nearly in every sentence in which they ask you whether it supports and the prediction and all that. You can write that in nearly every such answer for support and prediction. So chimpanzee highest genetic variation but endangered. Minke whale has least genetic but is least concern. Genetic variation same but conservation status is different. Then you gave me an example of it which I just told you about, that which one are the same. The whale, this one is the same, and the lion is the same, but their conservation status is different. So you could give me an example of any of these which you wanted to. So this is the one which I'm talking about in the example. Then gorilla critically endangered but has the higher genetic variation. And then the data is limited to only 7 species. So the last point was data limited to only 7 species. So I've told you this, that any case in which you need to support and all, you can always talk about this is that the data is of, if only a few species have been talked about, or in any other situation, then you can't sort of make it a general rule for the whole world and say this is the rule for everything, for trees, for animals, for plants, for insects, for any other species, monkeys, chimpanzees, oak trees, mango trees, Anopheles mosquito, fruit flies, green frogs, anything. No, you can't say that. You can't generalize because we've only got this data on 7 different species.